everybody. This is session number 5. Session number 5 will take you 60 minutes to complete, and this is about life sciences. So this is biology. We'll spend some time talking about cells and organelles. So we'll talk about the difference between eukaryotic and prokaryotic cells and kind of like how do we differentiate between those two. We'll also talk about cell division and some genetics. So there's some inheritance problems in there, like Punnett squares and Mendelian inheritance, non-Mendelian inheritance. You probably heard it in one of the recordings, but the biggest part of this test is physical science, which is why we spent 4 full sessions talking about some concepts in physics and chem. This life science is second, so biology is also a pretty good chunk. We've heard from previous enrollees and previous residents that the biology section is a lot of memorization. So make sure you know your verbiage, vernacular, make sure you know your bio terms, make sure you know your taxonomy, make sure you know the different organelles and their functions. So we'll talk on some of these things in sessions 5 and 6. I just wanted to remind you before you embark. That's all I have for now. So I'll see you at the top of session number 6, and happy studying.
Cell wall.
Good, so I got cell wall. So what is the function of the cell wall, y'all?
Is it like, Lord, I haven't got this far in my study and I'm still in like chemistry and physics.
I'm gonna review all this stuff. I just want to see where we're at. Yes, ma'am, Ms. Simmons. Cell wall is protection and structure. So plant cells need to be a little bit more rigid, so they have that cell wall built in there. Good. The other one I'm seeing is chloroplast. That's usually the easiest one for us to kind of get.
That's what I meant, chloroplast, not cytoplasm, chloroplast.
Chloroplasts are only in plant cells, and what do chloroplasts do?
Oh, good.
Doesn't change sunlight.
Huh?
Yes, I'm like, I heard photosynthesis. That is the big part right there.
Yeah.
Good. And then Bryson said green. Thank you, sir. We appreciate you. Sorry, I'm gonna always talk trash to him. I'm so sorry.
And we need to talk about food, greens.
Oh yes, Lord. Anybody eat some greens yesterday and want to bring me some?
Oh yes, yes, yes.
That's pregnancy talking. Sorry, y'all.
You good.
The other one that people typically tend to miss is our big guy in the center over here, and that is the vacuole.
Oh, the vacuole.
So in plant cells, you're going to find a gigantic vacuole. You do see some vacuoles in animal cells, but they're never this large. So in other sessions, I have not kind of spent time going through each of these organelles, but I do want to spend the time doing that tonight. So let's jump in a little bit, and then we'll take some time to practice these, because I did hear back that there was more on like the structure and function of organelles.
They asked about the chloroplast, like they had like their own little diagram of it, and then they had like the pieces stacked, so it had it labeled. And then it would like ask you, like you about to tell, like what was the function of like the stroma. I had something about the stroma or something like that.
Okay, I'll add that in there too then. So a little bit more on the plant cell function. I focused on animal cells tonight because I thought they might ask more eukaryotic, but just in case, I will send some notes on plant specific. Okay. So let's talk about a couple organelles. So before we talk about the organelles themselves, we need to talk about our 2 different types of cells. So first on the left, you have your prokaryotic, simple, single-celled organisms, and you have your eukaryotic. Those are our multicellular, multifunction, more complex organisms. So to kind of think about them, okay, we'll go back. So my screen is not loading, that's okay. Hold on, guys. I have a UVerse, so some days things just don't work. Hopefully it'll work now. Here we go. All right. So when we think about our prokaryotic cells, we want to think about our simplest type of cells. We will talk about like the different phylums and kingdoms and all that good type of stuff. And you'll see that one of the kingdom names for bacteria is the archaea. And that literally just means like very simple bacteria. The word breaks down to prokaryo, which means before nucleus. So these cells typically do not have nuclei. They don't have all the different organelles that we're going to see in our eukaryotic cells, are more complex. They usually don't have those membrane-bound organelles. So they have a couple things, but very, very simple. Our eukaryotic cells are, so if you break down the word eukaryote, true nucleus, so these are the ones with the nucleus. So things like fungi and our protista, which are not bacteria, but are like one step above. So you're like algae-like creatures. Plants and animals are all eukaryotic cells. They are far more complex. They have chromosomes, so we have DNA involved here, and we also have our different membrane-bound organelles. So we're going to focus on eukaryotic cells and the organelles that they contain, starting with the ones that all eukaryotic cells have. So whether you're plant, plant, animal, fungus, protista, whatever, you all have these 4 things in common. So first thing would be DNA. All eukaryotic cells have DNA organized as chromosomes. The amount of chromosomes you have will differ depending on organism, but all types of eukaryotic cells have that DNA coiled up into that chromosome structure. All eukaryotic cells have cytoplasm. So that is the jelly-like thing that fills up the cell where all the organelles are floating around. They also all have ribosomes. So the ribosome is an organelle that is where you get your proteins. So cells have to be able to produce their own proteins to live, and all those proteins are synthesized in our ribosomes. Also, all eukaryotic cells have the cell membrane, also known as the plasma membrane. Your test may ask you about the structure of the cell membrane, so you definitely want to know a couple key terms like phospholipid bilayer, which means it's a double stacked organism where it has these 2 fatty heads on the end and tails that go on the inside. The heads are hydrophilic, which means they like water, and the tails are hydrophobic, which means they don't like water. That's why the tails are on the inside. The plasma membrane is semi-permeable. Hold on, I saw something in the chat. Oh, am I back, Ms. Simmons? Am I breaking up for everybody?
You're not bad for me.
Okay. I will try to slow down a little bit too. I could be talking too fast. The plasma membrane is important because it's semi-permeable. So it's supposed to let some things through and not everything. So they have the processes of osmosis, you have diffusion, that's where all those things come into play. I'll send some notes about like active and passive transport. I don't know if the test asks you about that, but I will send the notes that I'm gonna send you guys from where I'm teaching from has everything like detailed, broken down step by step. But just remember, cell membrane is present in all eukaryotic cells. Plant cells have that extra little layer of protection on the outside, but animal cells just have the membrane to allow for osmosis and diffusion for things to pass through selectively. Now let's focus on some of the differences. So when we talk about our eukaryotic cells, first thing that all eukaryotic cells have are our nucleus. Okay, some of our prokaryotic cells do not have the nucleus, and some of our, I'm stuttering today, sorry y'all. So most people know that the nucleus is our powerhouse. This is our control center. This is where your DNA is located. So all the things that make your cell work, all the reasons why it can function, all the things that code you for you are located inside your nucleus. As a forensic science teacher, we spend a lot of time teaching kids that like the reason why you can't get, they ask this question sometimes. They might ask you about like blood and like why you can't always get DNA from a blood sample. And that's because you only have 1 copy of nuclear DNA inside every cell. So if that nucleus is broken down and the DNA has broken down too, you no longer have that sample, which is why you really can't extract DNA, nuclear DNA, from degraded samples. However, you can extract something else, and that is mitochondrial DNA, which is the one that some of us aren't so familiar with. So while every cell has 1 nucleus, they all have many mitochondria. The mitochondria is the powerhouse. They produce our ATP, that's the energy that keeps them going, but they also have their own type of DNA that is linked to your mother. So all of us have mitochondrial DNA that has been passed down from the maternal side of our family. Ladies, we will continue to pass that on. Gentlemen, your children will have the mitochondrial DNA of their mother. So you can use mitochondrial DNA in degraded remains to test for inheritance. So if you find bones, or if you find really degraded blood, semen samples, you can test for mitochondrial DNA because there's more mitochondria around than there are in a cell. Also, you have your vacuoles. So it's important to know that the size of a vacuole depends on the function of the cell. So because animal cells are able to, they have that semi-permeable membrane and water can pass in and out. The vacuoles are very, very small. They don't need to store much water. Plant cells have that big old cell wall sitting on the outside, so they have to store water for much longer, which is why their vacuole is much bigger. It takes up a lot more space inside a plant cell than an animal cell. Lysosomes, sometimes people call them trash cans or pickups. Some people call them the wrecking balls. Their job is just to digest other things that are not supposed to be in the cell. So if a cell part is damaged, if there's been some waste byproducts, it's the lysosome's job to kind of clean all of that up. The endoplasmic reticulum, there are two types. You can have the rough and the smooth. Either way, overall, it's just a series of interconnected folded membranes, and its job is to kind of transport. It's like the assembly line. So the rough ER is covered with ribosomes, the smooth ER is not. But both of them, they just kind of transport proteins and molecules all around their cell for synthesis. And lastly, in our eukaryotic cells, all of them also have a Golgi complex or a Golgi apparatus. Okay. And this is the guy that packages up your proteins. So once they've been synthesized by the ribosomes, they get sent down the ER to the Golgi, they get all packaged and they go wherever they need to go in the body or in other cells. Questions on these guys? I really like GIFs, y'all, so please don't mind me. They make me laugh. Now, there are 3 other organelles that are only found in either plant or animal. So the first one is a centriole. Centrioles are only found in animal cells. Okay, so you don't find these in plant cells, and that's because centrioles help with cell division, mitosis and meiosis process. You don't have the exact same mitosis and meiosis in plants, so you only find centrioles in animal cells. The ones with the double asterisk, those are only found in plant cells. So we kind of already talked about our good friend, the chloroplast, that helps with photosynthesis. That's what gives you the energy that the plant needs, since they can't kind of actively go out and consume food for themselves. And then also our plant cells have that cell wall for structure, rigidity, and protection of the cells. Cell walls are made of something called cellulose, just in case they ask about that part. Not sure if they will, but it's good to know that cell walls are made of cellulose. And that was the majority of our cell organelles in 7 minutes, 6 minutes. Let's practice a little bit. So we have cells. Everybody knows that part. In order for us to keep existing, those cells have to keep dividing. So if we know that we have 2 different types of cells in our bodies, we are eukaryotic creatures, we have animal cells, so we have somatic or body cells, and we have our gametes, which are egg and sperm, our sex cells. The reason why everything in your body keeps going and keeps focusing is because those somatic cells keep dividing over and over and over again through a process called mitosis. Your sex cells go through meiosis, which we'll talk about in a second as well. So real quick, before we get to mitosis in particular, the test likes to draw a lot of compare and contrast between mitosis and meiosis. So I'm gonna bring this slide up again just to make sure we understand, but starting at the beginning, mitosis only happens in body cells. Meiosis only happens with our sex cells. In both of these cells, you're gonna start with what we call diploid cells, which means they have 2 chromosomes in them. In mitosis, that diploid cell is gonna go through 1 division. In meiosis, it's going to go through 2 divisions and produce haploid cells, which will be half cell. Okay. So before, I'm going to throw this slide again, but I know some people are taking notes. So before I keep going with like all these vocabulary terms, I wanted to make sure we know the difference between diploid and haploid, because that's important to understanding division. So again, diploid means that each cell has 2 chromosomes. If we think back to bio, we know chromosomes are those X-shaped structures. Chromosomes are made of 2 chromatids that are attached together. So the 2 halves of the chromatids make 1 chromosome. That means you have 23 pairs of chromosomes with 46 chromatids, right? So a human gamete, a haploid cell, only contains 23 chromosomes, like 23 itself. That's how we end up with our 46, because we get half from mom, half from dad, half from the egg, half from the sperm. Right? This is review. So let's talk about mitosis, because this is where some people, if you haven't taken biology in a little while, might be a little rusty on this process of division. Whether we're talking about mitosis or meiosis, we start with interphase. So both of them start with here in interphase, which is the longest part of this division cycle. In interphase, the cell is kind of doing its thing. The cell is making energy with its mitochondria, it's processing, it's doing whatever specialized functions it has to do, and it's also preparing its chromosomes to divide and create the next set of cells. So when we talk about mitosis in particular, we're going to hit 4 phase, and we like to remember the phases by PMAT. P-M-A-T is the order. So starting with the beginning of mitosis, we're going to go through prophase, metaphase, anaphase, and then telophase. Prophase is when you have spindles that start to form. Metaphase, which is usually the one that's a little bit easiest to remember, meta meaning middle, is when the chromosomes line up down the center. Anaphase, they are pulled apart. And then telophase is when you start to get 2 separate nuclei, and then cytokinesis is when it pulls apart. So prophase spindles, metaphase, they line up the chromosomes. Anaphase, they're pulled apart. And then telophase is when we have our 2 new cells. Remember again, over here on the upper corner is interphase, which is just the cell doing its normal day-to-day things. You have its chromosomes appearing, then they're duplicated, then they start to get ready for their separation. So interphase is when the chromosomes actually are duplicated. The whole cycle is when they're pulled apart to make our 2 new cells. Now, when we talk about prophase, metaphase, anaphase, telophase, the only difference really between mitosis and meiosis is that this whole PMAT thing is going to happen twice in meiosis. So if I look at them side to side, on the left I have mitosis going through the stages we just talked about. Prophase, metaphase, everything lines up. Anaphase, they get pulled apart. In telophase, we have 2 separate cells. In meiosis over here, you'll see meiosis 1, we have prophase 1, metaphase 1, anaphase 1, and here's where the difference lies. At the end of anaphase 1, you're going to end up with 2 different haploid cells. So each of these guys will have 23 sets, their own 23 chromosomes that need to be divided in half again in order for us to get our human genetics and variation. So our gamete cells, the sperm and the egg, have to divide twice in order for us not to have too many chromosomes. So that's why it's going to go through it all over again. Now here's where they like to kind of get people on questions on the test. Well, you know what? Let me back up for a second. Let's look at one more side to side just to make sure we understand. Okay. Again, just to review, mitosis is just in the body, 1 division that creates 2 genetically identical cells. So mitosis gives you identical cells. You want your body cells to be identical. It would be weird if your skin cells all of a sudden started producing different genetic variations randomly. That's where our mutations come from. The reason why we have the 2 divisions in meiosis is to increase genetic diversity. So if you have, like, most of us have some siblings, you can see that if we didn't have meiosis 2 the second time, you and all of your siblings would probably look like clones of each other because there would be no diversity between your parents' genetic cycles. When we start thinking about ecology, which is also a piece of this life science thing, we remember that we need genetic diversity. If we think about like habituation and survival of the fittest, we need this diversity to kind of weed out weak genetic links down the line. How are you guys feeling about mitosis and meiosis? Do you want a little bit more information, or do you want to try some practice problems? because I have a short video in case we want a little bit of a clearer explanation. And if you're feeling rusty, it's only about 6 minutes long. So it's up to you guys if you want me to show it on here or if you'd like me to email it out and we just try some practice problems. You let me know. Email?
Yeah, that's fine. Email.
I see some nodding. So everybody's okay with me just emailing this one out?
Yes.
Okay, so I will send this out in our email this evening. These guys, so for anybody who does end up teaching life science or biology when you guys are in your classrooms, because we're going to speak that one into existence, these guys, they have a whole bunch of videos that are like really cute and kid friendly. Like the animations are really simple and they break things down very, very easily. So I would definitely look into their videos to show your kids if you end up teaching life science or biology. Their videos are great.
What videos did you say?
It's right here. I forget what their names are. The Amoeba Sisters.
Okay.
And so they're all animated videos, but they're all really, really cute, and they go through the entire biology curriculum.
All right. Thank you.
But I'll put this in the email I send out tonight. So let's look at some questions. Which of the following diagrams best represents a cell in anaphase 1? So think about that clue right there and what we just talked about in mitosis and meiosis. I see some stuff in my chat box. Okay, I see some B's and I see some D's. So I'm glad. Which ones were we able to eliminate by process of elimination?
A,
A, because A is actually what phase?
metaphase.
Excellent. A is metaphase. They're lining up in the center, and we can also get rid of what?
C,
C. C is what phase, guys?
doing its regular.
Almost. This is actually right before they separate. So this is the T. This is telophase.
Okay, gotcha.
So now we've got B and we've got D left over. Now the key here is it's talking about anaphase 1. So would anaphase 1 be mitosis or meiosis?
Meiosis.
Meiosis. So you're looking for the one where you have them being pulled apart before they separate in half. So the answer here is actually D, down here at the bottom. This one would be regular anaphase for mitosis. However, this one at the bottom is anaphase 1 for meiosis, because we have just 4 full chromosomes being pulled apart instead of these guys right here are about to be pulled in half identical. Like, let me say this the right way. These guys right here are about to line up. So actually this one's probably prophase because they're about to get in their line. This one right here is before the big dense chromosomes are separated again. So these would need to go through another division in order for them to produce correct cells. So using this same image, I want to come up with another question because there's another way that they'll ask you this, and it's here. So it says, which of the following diagrams best represents a cell that, when finished dividing, would result in the production of diploid, genetically identical daughter cells? So which of these would give you identical daughter cells? Good job, baby. So I've got a couple answers coming in my chat. So some people are saying B, but guys, remember this one is probably prophase, which is the beginning. So you're looking for a little bit later on in the cycle. We already said it's not going to be D because D would be in meiosis. And meiosis give you identical cells? I see people shaking their head, nope. So it'd be the one stage that most likely represents mitosis. And in that case, the answer is A. Okay. So here, even though C looks like when it separates, we're gonna probably get 2 identical cells, we are not 100% certain. Some of this DNA could swing to the other side, or some of this DNA could swing down here. So when it says best represents, C could give you 2 identical cells, but the one where it's most likely to happen would be here in A. These guys will be pulled exactly in half and give you 2 identical daughter cells, because this is the metaphase of mitosis, not meiosis. So this is the type of way it likes to get you. They'll give you the same picture and ask you 2-3 different questions about the same image and try to get you to kind of second guess yourself. You guys are so smart, so please don't second guess yourself. I saw some people put one answer and then go change it after a second. You were right the first time. So, y'all want to hear another corny joke before we go on to some more genetic stuff?
Sure.
Everybody's like, no, oh my gosh, stop telling me jokes. But I really like jokes, and it breaks the circle. So what does a cell say when you step on it? Ow, mitosis. Okay, sorry. I teach through really corny jokes, and like my kids just know that I'm a corny teacher. Whatever, it's me. Sorry, y'all. All right. So let's talk a little bit more, and it's going to be tied to our cells, but let's talk about some genetics. We're going to work with some Punnett squares in a second. Before we talk about genetics, let's see how we do with the question like this. A briefing question. It goes about our cell division, but it's also about something you probably already know. Right, good job, guys. I see the answer coming up in the chat. Everybody got this one right. This one is B. This one is B, Down syndrome. The key here, and what most of us already know, the other name for Down syndrome is what? You can say it out or put it in the chat if you'd like to. Anybody know? There you go, trisomy 21. Tri meaning 3, and that means you've got 3 chromosomes on the 21st part of your karyotype. So this person has a relatively normal karyotype except for the 21st chromosome. They have 3 instead of 2. So this person would have Down syndrome, trisomy 21. So, yeah.
I'm just curious, how do I like type answers?
Okay, if you, I don't know what the screen looks like for y'all, but there should be, is there like a little help bar somewhere that says more?
Yes.
So if you hit more, you should see a button that says chat.
Okay.
In the chat box that's where you can type anything. You can type to everybody else or you can type to me.
Okay, thank you so much.
You're welcome. Not a problem. All right. So let's talk a little bit more about our chromosomes. We already said that our chromosomes exist in pairs. So we have 23 pairs of total of our 46 chromosomes. The 23rd pair are our sex chromosomes. So females are XX, males are XY. Genes that are carried on the X chromosome are said to be sex-linked. So that's things like male pattern baldness and color blindness. So more males are colorblind and more males have baldness, because if they have that trait on the X chromosome, they don't have another X chromosome to balance that out. Make sense? Mm-hmm. So even though we like to say sex-linked traits are mostly contributed to the male, it's not attached to their Y gamete, it's attached to their X. If they only have one weak X gamete, they don't have another one to balance it out. So that means we as women can be carriers of a weak X chromosome, be recessive, and then you can start to see some of those traits expressed in your children if you have boys. So like my son has a pretty high likelihood of being colorblind. All of my brothers are. So that means I probably carry, one of my exes may carry that colorblindness trait, and I could pass that down to a man. So let's talk about that whole passing down thing through our good friend Mendel. So for my middle school people, they like to ask you about a lot of names, and Mendel, this genetics and inheritance thing, and he has 3 major principles. If you've ever studied biology, if you've ever done a Punnett square, you already know these principles, but just in case you didn't know the names for them, here they are. So first thing he said is that we have dominant and recessive traits. So we all have chromosomes. On our chromosomes are genes. All of us have different versions of those genes called alleles. Are dominant, some alleles are recessive. If you have a dominant allele, that is what's expressed, and the recessive one stays recessive copies. That's the principle of dominance. The principle of segregation, it says that you don't pass down all of your alleles to your offspring. You only pass down half. And then independent assortment just says we have no way of figuring out which alleles you're going to pass down to your offspring, even though genetically speaking, people are trying to do that now, because now you can genetically modify a baby, which is creepy. Okay. But again, if you've ever studied anything about genetics, or if you've ever done a Punnett square, you already know these principles, but this is just where the names come from. Questions here? Okay. Oh, puppy. Hi, puppy. Sorry. All right, so let's start to make some Punnett squares. Am I moving?
Just a little.
Oh, I'm sorry. Let me slow down. My bad. I'm also going to do a lot better this week at sending out the PowerPoints ahead of time. I was so busy working on those Praxis guides that I didn't have time to do this other part. So now I can do that and get my life together. I'm sorry. I will also send out the recording of this probably tomorrow, because it takes about a full day for them to get synthesized.
Okay.
Good.
Thank you.
There are 2 types of inheritance patterns in genetics. The first type is based on Mendel, so we call it Mendelian inheritance. And then we have non-Mendelian inheritance. We're going to run through some examples of each of these, and then I'll give us about 10 minutes to practice problems in smaller groups, and we'll check in. So in terms of Mendelian inheritance, this is the one that we're probably most familiar with. In Mendelian inheritance, you can solve all of the problems with our Punnett squares. So Mendelian inheritance is just our dominant and recessive alleles. So let me I apologize in advance, y'all. I'm gonna try to draw on the screen with my fingers. My nails are kind of long, so we'll see if we can make this work. So in our example problem, it says we have a tall pea plant that is dominant to short pea plant in terms of height. It wants you to cross a pure breeding tall pea plant with a pure breeding short pea plant and give the genotypic and phenotypic ratio. It's a bunch of words to say that you have a tall plant and a short plant, and what do their babies look like. So if I'm making a Punnett square, I'm going to draw a 4x4 box. And just for the sake of this exercise, I'm going to use big H to represent tall and little h to represent short. If it says one of my parents is pure breeding, that means it's 100% tall. So it's got 2 big H's. And then my other one is pure breeding short, so it's got 2 little h's. When I fill in the Punnett square, I'm going to put whatever the dominant allele is first next to the one. So I'm going to do top and then the side. So I've got an uppercase H from one parent and a lowercase h from the other parent here. And because we have 2 homozygous creatures, all of the babies are going to be the same. So for the genotypic ratio, the genotype is 100% HH, right? They all have 1 big H and they all have 1 little h. In terms of the phenotype or what they look like, are all of my plants tall or short? Tall. They're 100% tall. None of my plants can be short because none of my plants have inherited recessive allele here.
So is pure breeding and homozygous the same thing?
Mm-hmm.
Okay, just making sure.
Everybody cool on that example?
Yes.
So this is probably where people are most familiar with a monohybrid cross. Mono meaning we have one trait on both sides. In Mendelian inheritance, you can also end up with a dihybrid cross. And for the sake of time, I'm gonna put the cross up on the board for you because it's gonna be a bigger one, which means you have basically two Punnett squares at the same time. In this one, it says we have guinea pig's fur that is black fur is dominant to white fur, and rough fur is dominant to smooth fur. And it wants you to cross two heterozygous black, rough guinea pigs. So in a case like this, we have to look at two traits at the same time. Instead of us doing two different Punnett squares, we're just gonna make one big 4x4. To do that, we also have to think about what the genotype for the parents are. So if we have black fur being dominant to white fur, oh, here we go. There we go. Sorry, y'all, it's not working. So if one of them, that means big B would be a black one, and little b would be a white guinea pig. When it talks about the rough and the smooth fur, I'm just going to use R's because I can draw that out bigger. So big R would be two rough fur, and little r would be smooth fur guinea pigs. However, this wants us to cross two heterozygous guinea pigs. So that means the genotype for each of our parents is going to be this.
Yeah.
Right. Each of the parents, because they're heterozygous, each parent will have a dominant and recessive of each of the alleles. So when we do a dihybrid cross, which is all four of these guys at the same time, it's going to look like this one. Right. What you have to do here is you have to lay out all possible examples for these parents. So because they are heterozygous, you have to put out everything. So they could be 100% dominant, a dominant recessive, every possibility that you could possibly have is what's laid out in a dihybrid cross. It's the big one.
Gotcha.
So when you look at their babies, this baby has what phenotype? What color is it, and is it rough or is it smooth?
It's black and it's smooth.
Remember, rough is dominant to smooth, so it's black and
Rough.
black and rough. And so most of their babies are going to be black furred and rough.
Mm-hmm.
What's this baby?
Black fur and
It is
soft.
black and smooth.
Good.
Or smooth.
There you go. We got this, right? This is what we're gonna practice these in small groups. So we're gonna run through the examples, and then we should still have about 11 or 12 minutes to practice in smaller groups. So in non-Mendelian inheritance, there are a couple different types. So first you could have something like codominance. In a codominant situation, both alleles are equally dominant, so both are expressed. So that would be something like a roan cow. They are brown and white, and it's because in roan cows, the brown and white are equally dominant, so both traits are equally expressed in the animal. Same thing like calico cats. The colors are co-dominant to each other, so you end up with this beautiful multicolored cat. Incomplete dominance is similar to codominance. However, here, neither allele is quite dominant enough, so you usually end up with a blend between the two. So codominance, you end up with like a spotted mottled appearance. In incomplete dominance, you're gonna end up with a blend, kind of like in these snapdragon flowers. So in snapdragons, the pink and the white, neither one of them is fully dominant. So if you cross the two, you end up with a pink, because they're just gonna form a blend instead of making it purple and white if it was a co-dominant situation. Another form of non-Mendelian inheritance is multiple alleles, and that just means you have more than 2 alleles that exist within the population for that given trait. Did I put a picture up? Yes, I did. So I'm gonna go to that example in a second. Easiest way to think about multiple alleles is blood type. So all of us have a blood type, A, B, and O. If you are type A or type B, your blood type is determined by 2 alleles. So you could be BB, which means you have both the dominant, or you could be BO, which means you have the dominant and the recessive. So for me, I'm type A. My son is type O, and his father is type B. In order for my son to be type O, that means both I and my son's father have to be AO and BO for us to pass down the O trait for our son. And that's just multiple alleles. So we carry specific alleles, kind of like regular Mendelian inheritance. It just means that one trait can be expressed in 2 different ways. So an example problem would be like this one. In blood typing, there's this right here, IA and IA little I, is just the same thing as saying big A, or big A, O. The lowercase I represents the O allele. So if it wanted you to, this problem comes from a released Praxis as well, and it says to cross a heterozygous type A female with a type O male, what are all the possible blood types of the offspring? At first, it seems like you're gonna have to do a big old dihybrid cross, but luckily because it tells you the mom is heterozygous type A and the father is type O, you can do a regular 4x4 Punnett square to solve this problem.
So it can be A or O.
So if I draw it out, the mom is heterozygous type A, so that means they're gonna be IA, and then a lowercase I over here. The father is type O, and the only genotype we can have is lowercase I. Same thing as saying O. So when I finish the square, this first baby, what's the first baby's blood type?
A.
Type A. This baby is
O.
O. Y'all don't need me for this part.
And A and O.
A and O. 50% A, 50% O for these children. Cool? All right. One more thing before we do our practicing. Oh. Another type of codominance is sex-linked. So we said in the beginning of this part that sex-linked traits are attached to the X chromosome. So in this case, it says colorblindness is a recessive sex-linked trait. A colorblind man has a child with a woman who's a carrier for colorblindness. What is the chance they'll have a colorblind son? You're gonna set this one up again, like a Punnett square. And for the sake of time, I'm just gonna throw up the square here for us. So all we did that was slightly different is we gave the parents, parent X is XX and XY. XY is the dad, and we put a little lowercase b because they are colorblind. So remember, sex-linked traits are attached to an X chromosome only. So that means the dad's genotype is X little b Y, showing that he is colorblind recessive. It says the mom is heterozygous, which means one of her X's has the dominant and one of her X's has the recessive. And when we do for their trait, do they have a chance of having a colorblind son?
Yes.
Yeah, what's the chance?
25%.
Just 25%. Down here in the corner, when we crossed our X's and our Y's, we have an XY child, which means it's a boy, and one of them got the lowercase b from the mom. So we do have a 25% chance of having a colorblind child, AKA myself. Hopefully, we'll see. Last, for non-Mendelian, and I know there's a lot of stuff here, so I'm gonna make sure I send out all these notes. The last one is just called polygenetic inheritance. The prefix poly means many, So polygenetic just means that some of our traits depend on many genes. So things like our eye color and our hair color and our skin color, that's not just 2 alleles that give you that. It's a mix of many genes at one time. Even things like the size of your feet. Foot size and hand size is polygenetic. It's determined by a bunch of different genes, not just 2 alleles from each parent. Okay. So let's do some practice. I'm gonna split you into small groups, and I'm gonna put the link in the chat before I split you. There are a lot of problems in the practice, so I am not expecting you guys to get through all of them. There's some that if you want to take some more time to practice after, if you want to send them to me so I can check them, if you want to call in and we want to check them one by one. So in the 10 minutes, I'm probably only expecting y'all to get through maybe two, 3, 4 of the problems. And then we're gonna come back together and try a couple more just to make sure. I know some people don't like small groups, I understand. But it's one way. I'm not always the best at explaining things, as I'm sure you guys have heard many times. I stutter a lot. So this is a way for y'all to kind of get some information from somebody else, because I bet you there's people in here that are way better at genetics than I am. This link is a read only, so if you want to make a copy for yourself to keep, you most certainly can. It's also something I will attach to our our files tonight for the people who are driving right now. So let's see. We have 12 people, so I'm gonna make 3 rooms with 4 people each. You guys should have the notification, and I will be popping in and out of all of the rooms to meet you. Josh, I did it to you again. Oh, you're muted. It didn't send you a thing again, huh?
It did not.
I don't know why it always does that to you. Well, if you want to pull up the document, we can look at it together.
Okay.
And then I'll pop in and out the rooms.
All right, so this is gonna be Punnett square. Vampires, long things are dominant, so I guess L and 2 heterozygous long thing vampires. So big L, little L again.
Oh, hold on 1 second. Somebody asked me for help, but yes, I'll be right back.
Okay. It's gonna have long things, and then it's gonna 100%. LL or big L, little L. This is 3, and then 3A. B werewolves. Ability to shift when the moon is not. Is tested. Grace, who can ship at any time, and Sam. Okay, so one is not full, so a little for you. Okay.
Gosh, I'm sorry.
Shipped anytime. No, you're fine.
Something is. I have one participant who keeps getting separated from. He gets separated from his audio every time, and I don't know how. Okay.
SMH. All right, so this first one, this 3A. So like it said, 2 heterozygous long-finged vampires. So like each one of them is a capital L and a lowercase L.
Yes.
And then like if I make the Punnett square, I have like at the top, I have the 2 capital L's, and then I have the or 2 lowercase L's on the side.
So that you would have on the top, you'd have an upper and a lower, and on the side, you'd have an upper and a lower. So on the top, you should have one complete parent, and on the other side, you should have the other parent.
Okay. So I would have LL, 2 capitals. Oh, actually, okay. Then the first one and the second one are both big L's, 2 of them, and then it's a capital L and a lowercase L, and then there's a 2 capital L's. So its phenotype would be 75% long things.
Yep.
And I guess 25. I guess you put 25%. Well, no. Well, wouldn't it be 100% long things or no?
No, because the last little baby would have 2 little L's, so that last one would have short fangs. That would be the only one with short.
Okay. Yeah. Okay. Yeah. And then so the genotype would be 75. 75. LL big L's, and then 25% big L little L.
I think so. When I did it, I got 25% big L, 50% big L, little L, and 25% little L, little L.
That's correct. That's correct. I had my, I wrote it right, and then I don't know what I did over here. But yeah, that's correct. That is correct. Yeah, 50. Yeah, 50, 25. 20, yeah, 50, 25, 25.
Yes.
Yeah.
All right, let me check on one of the rooms. I'll be right back.
All right.
Okay, let me check on the second one.
One shipped on a full moon. And then the other one only shifts like it ship anytime. So
in just a few weeks, I was confident enough to have an entire conversation in Spanish. Las gambas have been deliciosas. Gracias.
shift at any time. When the moon is not full, it's recessive. They would.
Sorry, you all right, Josh?
Yes, I think so. Like, I'm trying to figure out, like, 'cause sometimes these questions, like, they add information that
Yeah.
I don't know if it's like necessarily pertinent to the question.
Not at all.
But so, like, this question, like on 3B, I just feel like that information about the mother is irrelevant. I don't know if it is or not, but it just sounds irrelevant.
Talking about Sam's mom.
Yes. Or is it relevant?
So I think what they were trying to get you to understand is that Sam is heterozygous.
Okay.
Because if his mom could shift at any time, hold on, wait. Yeah. The ability to shift any time is recessive. So that means his mom would have to be fully recessive, but he would only have one copy if he could only shift during the full moon.
Okay.
And then Grace can shift at any time. So that means she has both recessive alleles. So they're trying to tell you that Grace is like, so say we use, I don't know, S for shift. Grace would be,
S.
Yeah, but Sam would be big S, little s, because he can only shift when it's a full moon.
Yes. So they can have a child that no, it's not recessive. Hold up. Shift anytime. No. Yes. So shift anytime is recessive. Yeah. So they can have a child that could shift at any time.
Yeah, I got 50% chance.
Okay. Yes. That's what I got. And then I got like one where like they only shift during the full moon, and then they have. Well, I guess they have two. They have two that, a half and half here.
Yeah. Okay, let me tell everybody to come on back.
Rockville, North Carolina.
Oh, they coming back. Welcome back, welcome back, welcome back. Brian, don't forget to switch your video back. There you go. Welcome back, my friends. As you're coming back, I have another question for you. So I have a question to check in and see how you're doing. Got a couple people, one of my rooms is still on their way back. So as they're coming, please try to start puzzling through this question on my screen with the cystic fibrosis. Wait, we on something else now. All right, welcome back. So just to check in and see how we're doing, I heard some really great conversation as I was popping in and out of the rooms. It says cystic fibrosis is genetic autosomal recessive disease. If a woman with cystic fibrosis has children with a man who does not have cystic fibrosis, is not a carrier, what fraction of their offspring will have cystic fibrosis?
Fourth.
0.
Draw the square just to make sure. So I'm hearing the answers. Oh, opened up the wrong thing. Oh, where's my answer?
You can get this one.
Let's see. do. Autosomal recessive. So if she is autosomal recessive, that means does she have 1 or 2 copies of the allele?
2.
2. So I'm going to use D for disease just because it's easier for me to draw on the screen. And then the father, he does not have it and is not recessive. So what would his alleles look like?
DD.
Yep. And he would be capital or lowercase?
Oh, capital.
Capital.
Yeah.
Right? So if we cross all of their children, they will all be
The carrier.
Capital D, lowercase d.
yeah.
Mm-hmm.
So if it says genetic autosomal recessive disease, do their children have the disease?
No.
No.
No, they
No.
don't have the disease, but they're all what?
Carriers.
Carriers.
Recessive.
So they're all carriers. So 0 of their children will express this disease, but 100% of their children will be carriers of it.
And so if they married somebody that has it, their kids have a higher chance of developing it.
Exactly. So say one of their kids
Would be big D, little d, and then little d.
has children with somebody who does have a disease. So their first child would be here, but then they would end up with a 50% chance of their children carrying the disease.
Okay.
Excelente.
So the answer to the original question is 0.
Absolutely.
Mm-hmm.
All right. How are we feeling, guys?